So, for this next part of the conference, we'll be hearing from some of the members of our fellowship. One of the strengths of our community is, of course, the breadth of expertise across science, conservation, and animal care. And today we are very delighted to showcase just a small part of that incredible work. So we've got some more speakers for you folks. First, we will hear from long-standing fellow Edmund Flach, who will reflect on more than 60 years worth of association with ZSL. And
okay, yes, I can't quite keep up with Jim's number of years, but I worked out that, yes, I have been associated with ZSL for over 60 years. Going back over family photos, I was hoping to find one, when I was a really little kid, but the earliest I could find was 1966 when I was 8 years old, and yeah, that's me on the left, not looking very happy. But as for most of you, early memories were visits to both London and Whipsnade zoos, and just I even early on I loved taking photos, so I got the old Vulture here at Eland to Whipsnade and so forth. So that was an early love of the zoos. And then I sort of liked the idea of becoming a vet. I'd sort of read a number of books, including the sort of James Herriot books. And I thought, zoos and how could I get in? Being a vet might work, maybe. And during my course at Cambridge, you had to see practice, as they say, and so I wrote to London Zoo and was accepted for a 2-week placement. It was during December of 1981, I think that was. Yes, December 81. And a great team in the hospital as it still is, and very excited by the news of the donation that's going to see a new health centre, because obviously it's not changed much in the years since there, so it needs refurbishing. And then after working in general practice and then doing some overseas research, parasitology research, I was very lucky to be accepted as the veterinary officer at Whipsnade Zoo, and I worked there from 1992 to 2006, and it was just amazing. The variety of things I was doing, from obviously clinical work. They've made blood sampling a giraffe, doing postmortems, all sorts of things, including some overseas trips. And down the bottom, that was one of the greater one-horned rhinos that came from Nepal to Whipsnade. And I flew out to Nepal because we had to get blood samples to bring back and be tested for things like foot and mouth disease before the animals could be allowed in. So that was quite a nerve-wracking journey, I'll tell you. After 14 years doing that, I then moved sideways to become the pathologist, based at London Zoo, but still doing work up at Whipsnade. And again, an amazing opportunity. I mean, just every day was different, literally every day. It could be a mountain chicken frog I was working on, could be a rhino, where obviously you can't do that in a post-mortem room. There's just not room. So, you had to get a whole team in to do things like that, down to the potular snails down the bottom. It was just an amazing variety of things. So thoroughly enjoyed all of that work. But then retired in the end of 2019. And thinking, retirement, I'll take it easy and, then get involved in some things. And then, of course, COVID comes along in 2020. But after that, I did get back involved on a voluntary basis. I'm still working on writing up scientific papers on the work I was involved in, particularly on the pathology side. I'm still helping keep track of the pathology archives, many of which are still in these sort of box files in the attic up at Whipsnade and down in the basement at London. And, getting papers published. That was one on potular snail export, pre-export health screening. And of course, that's been a hugely successful overseas conservation project that ZSL's been a linchpin of. And just occasionally helping with things like zoo nights. And I even was in one of the first working groups weeding the chalk line at Whipsnade. I only did it once, but don't tell them. And there you go. That's a very quick snapshot of 60 plus years and still enjoying it. Thank you.
That's amazing. I love it. Now, Rajith Dissanayake will share insights from his work on palm squirrels and the Bombay Natural History Society Mammal Survey. Welcome.
Well, I'm very happy to be here with you today after 200 years of the ZSL. This is the first talk I'm giving since about 2009 when I sort of concluded my PhD at the Natural History Museum. As a country, India now has the world's largest population, and the wildlife has not done that badly under those circumstances. Now, as you would realize, birds have been worked on a great deal, especially the birds of Asia, amongst lots of other birds. And squirrels represent honorary birds. I mean, they are daytime creatures. They're diurnal, and they associate with birds. But relatively little work has actually been done on small squirrels. I mean, not just Indian ones, but around the rest of the world, especially when it comes to field work. A lot of museum work has been done, but very little field work. Most of my work was done by measuring the skulls of about 450 specimens from the collections at London and the United States. And most of my presentation will be actually hopefully giving the squirrels a chance to speak for themselves. So I've also done a bit of field work, especially recently. So this is the mammal survey, the Bombay Natural History Society Mammal Survey. 80% of the collection surveys of the collection, there were about 40 odd collection sites that they looked at in India, and 80% of these locations had funambulous palm squirrels. So you can see the 80 on, about the 40 odd collection sites on the map. The size of the circles represent how much work was done in each area. All the results were published in the Journal of the Bombay Natural History Society, and this survey was one of a kind. Not a single survey has been done that's quite so long, from 1912 to 1929. It was spearheaded by somebody called Robert Charles Ruffton, and it's very difficult to find a picture of him. But also the leadership was taken by Oldfield Thomas, whose picture you see. And Oldfield Thomas was one of the most prolific mammalogists in the whole world. He described vast numbers of species. So during the mammal survey, lots and lots of skins and skulls were pouring into London. And these scientists and volunteers were working on making publications. Some of these collections were also retained in the countries where the collections were made. So at the moment, Funambulus is a relatively small genus, and the name Funambulus has been recognized for this particular group of squirrels since about 1897. But actually, it's very difficult to tell squirrels apart generally. They have very few morphological distinguishing characters. So this genus is quite unique. First of all, it's quite difficult to tell this genus apart from some of the other genera in the same area. But this genus is very largely confined to the Indian subcontinent, and they're also stripy and they chirp a lot. Two of these species, the Pennanti and the Palmarum, are quite abundant, and you can't really miss them. And this genus is they're known as palm squirrels after the palm squirrel on the bottom right. And so it's called the palm squirrel. and the top one is called the northern palm squirrel. In the middle, you see Funambulus layardi that is endemic to Sri Lanka. And then on the left, you see Tristriatus that is endemic to India. And then on the bottom, you see two subspecies that used to be called Funambulus sublineatus, but thanks to my work, they have now been split into two species. One of them is called obscurus, and the other one is called sublineatus. We're calling the obscurus is called the dusky striped squirrel, and we're hoping to, well, I've named the Indian one has been named the pygmy striped squirrel. Many of the taxa that were described by the Bombay Natural History Society Mammal Survey have been subsumed as subspecies, and there is a potential for reviving some of these taxa. Now, once upon a time, Funambulus pennanti had been introduced to Australia, but that population has been eradicated because they were terrified of it spreading. But I managed to get 3 dead bodies from Australia for my research because it's very, very difficult to get specimen material from India itself. So this is Funambulus palmarum from Sri Lanka. This is the one from Sri Lanka. And I think that there could be, I think that the taxonomy of this particular species deserves further work. So this is the one from Sri Lanka, being its musical best in a kind of chorus. They start doing this, most Funambulus squirrels, especially these more common and abundant ones, they start doing this from about 6:30 in the morning, and they can do this at any time in the day. So this particular species is, as recognized, is found in southern India and Sri Lanka. So you can hear some others in the background as well. So they're all in a big chorus. It's usually the males who do this, we think. So there are hundreds and hundreds of these specimens collected, particularly in the London collection. The next one, this is quite an attractive squirrel. It's endemic to Sri Lanka, and it's called Layard squirrel, after Edgar Layard, who was sending specimens to India, and Blythe, somebody called Blythe was describing them, and he named it after Layard. So I think I'm the first person to have filmed them and kind of, since books were published about these animals in the 1930s, hardly anybody had touched them. So it was quite exciting for me to try to actually find them and to see if I could actually film them. And so I did get that far eventually. They were described as being living in thick, heavy forests with the tallest of trees. But then I found places where they're not supposed to be coming down, but there are places that they come down for food that people put out. So very, very shy. And this one is suppressed by Funambulus, the palm squirrel. If the palm squirrel and this animal are side by side, as it were, eventually the palm squirrel will outcompete this one. So this one is a very kind of wild and endemic animal that's not very well known. So here it is at sunset at Kalugala Reserve. Now, this one this is Funambulus sublineatus, sorry, it's called Funambulus obscurus now from Sri Lanka. And they call it the dusky, it used to be called the dusky striped squirrel, including the Indian species, but now I hope that people start calling the Indian one the pygmy striped squirrel, because the pygmy striped squirrel is the smallest one in the genus. And this one is slightly larger, but they are quite distinctive. The distinction between these species was recognized by the Bombay Natural History Society, but then the taxa got buried under one species, and so part of my work was splitting them into two. So this is it chirping as well in Candy. This one is probably closely related to lyre squirrel based on molecular evidence. I had to go and collect some blood samples from them without killing them. This is the jungle striped squirrel from India. This is endemic to India. And once upon a time, there was a subspecies of this. I mean, it's currently recognized as a subspecies, but there was a related species that was called Rufton squirrel. And I went searching for Rufton squirrel last year in India, but my conclusion was that Rufton squirrel is probably not a true species, or the true taxon, and actually belongs with this one. So, I mean, I was convinced that Rufton squirrel could be a real species, but after a lot of searching, it turned out that it's in its heartland of Coog. So this is in Coog. I couldn't really find something that seemed to correspond with Rufton squirrel, so this is the best I got. So this is quite this is one of the largest species in the genus, called the jungle striped squirrel. And they're found in forests and gardens in the Western Ghats. They've got quite pointy noses. So I'd just like to talk about how many species are recognized. And a lot of work these days is done based on DNA work, but it's actually very difficult to get hold of fresh samples from India, mainly because of legislation. And another approach is, if you've got two similar species, you have to tell them apart by the sounds that they make. So Gilbert White was one of the first to split warblers based on their sound. So at that time, they weren't called warblers, they were called wood wrens. And he identified that there were two wood wrens. We now know them as the willow warbler and the chiffchaff, and they were split because they look similar, but they sound completely different. I mean, there would have been little boys shooting them, and all the skins, all the dead birds would have looked similar, but when you start listening to them, you can tell them apart, and this could be a way of telling apart these, the members of this genus as well. So I'm sure that many of you might have heard this. This is the chiffchaff. And there used to be an exhibit about this in the Natural History Museum once upon a time. So you can understand why it's called chiffchaff. And then the next one, the willow warbler. And I think that this is a good technique to use for squirrel taxonomy. And we are finding quite interesting differences between some of these taxa. So the top image shows you what museum skins, kind of schematic drawings of museum skins, and you can see that they're very difficult to tell apart. At the bottom you can see Funambulus palmarum, and you can see two different morphs. And how can we work out how many species there are? It's actually quite tricky. So most of my research is based on morphometrics, but maybe sonograms could be a feature of this work. I think that at the moment 6 species are recognized, but I'm working on a paper mostly based at the ZSL library here on the 7th species. So there might be, I think there could be a 7th species that could be endemic to India, but I actually think that there are probably more species, and some of the currently recognized taxa could be split. So all my work is self-funded, so I have to thank my late parents and the organizations that I'm most closely associated with right now, at least as far as research is concerned, the ZSL and the Linnean Society, and I'm really grateful for the libraries. I've also made a connection recently with the Indian Institute for Science Education and Research at Tirupati. I work at Bombay College, and those 2 pictures are from my other research, and that's my social media handle. Thank you very much.
Excellent stuff. Definitely have a newfound love for squirrels. So we will now be hearing from Kat Taras, who will introduce the Central Asian Vultures Project and her fieldwork experience in Uzbekistan. Thank you, Kat.
Hello, I am Kat, and as just was said, I'm going to be introducing the Central Asian Vultures Project. The Central Asian Vultures Project is a partnership between various organisations within the region of Central Asia and also conservationists worldwide, united together to learn about and protect the 5 species of vulture found in Central Asia, all of which are declining in at least some part of their range. And it started with the endangered Egyptian vulture, where there have been amazing work around the Mediterranean flyway, looking at all of the different places they go. It's a migratory species. It breeds in different places than it winters. And you can see all of this information here, but then in this massive area of Central Asia where it breeds, we didn't even know in 2021 where they migrated to for winter. So we started tagging some vultures with GPS tags. And so let's jump into some experience doing field work, where in 2024, in the summer, we were gathering people from Uzbekistan, Tajikistan, Kyrgyzstan and Kazakhstan, along with experts from Bulgaria and Greece and Armenia who'd been tagging vultures for a while, and they also let me tag along. And so we got to, speaking of tags, tag 4 different Egyptian vultures, and gets like proper hands-on training in trapping and tagging and trying to count the vultures where they gather at dump sites in southern Uzbekistan. So, not easy work. The only shade there was the shadow of the vehicle we drove there in. And we were sort of based on a hill overlooking the dump site. And as it was getting towards the middle of the day, we were almost like sitting on the floor right up to the vehicle to try and get the last bit of shade. And that's not for us. It's stressful enough to be grabbed by strange bipedal creatures who want to give you fancy accessories without doing it in the blistering heat. So that's for their sake. Now, having lots of vultures, that's great. And to give you an idea of the scale, on one day in this dump site, we counted more vultures more Egyptian vultures than can be found in the entirety of the Balkans, in the same year. And the Balkan population, very rightfully, it's like declining terribly, has like had multiple like EU life projects, like funding and dealing with it. So I've given you a little bit of a chance there to see if you could spot the vultures. So let's see how well you did. And you can see, especially with the subadults, that sort of the darker colours, that they really sort of blend against the background of one of the lesser-known tourist sites on the Silk Route. And that's when they're still. Let's see them flying around, and they're mixed up with some ravens and some smaller birds, and so, yeah, that's really tricky. And of course, it is a rubbish dump, so fancy. It's the kind of rubbish site that also has like organic waste from abattoirs, which is what attracts all of the vultures. I have never been so pleased to have a terrible sense of smell. Really big asset in vulture work. And on one memorable occasion, like it's a working dump site, so one memorable occasion, we just placed out all of our traps, really exciting, and then a bunch of rubbish was just dumped on top of them. So we had some very brave members of the team who went and dug out most of them. There's probably a couple still out there. At least it's not technically littering. But it is worth it. I mean, getting to be so close to these fantastic creatures. I've got a really quick video there for you of releasing one of the birds with its fancy little backpack to go do some science for us. There we go, off he goes. And if you were wondering, we did find out where they winter. Most of them winter in northern India and Pakistan. Occasionally they go a little bit further. And we've started also tagging Cinereus vultures, which is great. And they seem to also be moving around, which they don't really do in Europe so much. And then here, just like some up-to-date information. So Aris was one of the very first vultures that were tagged back in 2021. And he was like juvenile in the nest, hadn't fledged, absolutely adorable. There are photos. And this is the journey that he made in March, just gone, back to Uzbekistan. And this year, he's just old enough to maybe start breeding. So that's really exciting to sort of see that journey continue. Now, if you would like to find out more, this has been 5 minutes, you can grab me around the place. I have stickers. I'll have to go get them from the cloakroom. There are some links here and a QR code. We especially have an exciting merchandise stall with really gorgeous exclusive artwork. So please come give us some money. We have so many different plans to do, like in terms of disease, in terms of the threats. We're really so much work to do, and funding is really difficult. So please support the project and follow us and see what happens next. Thank you very much.
Wonderful stuff. I will be pestering you for a sticker. I would now like to welcome Dr. Stephen Allain to the stage, who will explore who will explore the fascinating origins of Britain's midwife toads.
Trying to keep the time. Thank you. Okay, guys, so good afternoon. Good to see you all. You're slightly older than the general audience that I lecture to, because I'm a university lecturer at Anglo-Ruskin University, Writtle, and today I'm going to be talking about midwife toads. So, what we're going to talk about is where we know about midwife toads around the UK, where those populations are, but also, most importantly, where they came from. So, before I jump into all of that, hands up to those people in the room, of course, which of those of you knew we had midwife toads in Great Britain to start with? A handful of people, and I suspect that those people that aren't putting their hands up are people I've already spoken to or are aware of my work. So you kind of outed yourself there, guys. I apologise. So, where they come from, and doing a bit of digging in the literature, came across this record in Sir Christopher Leather's wonderful book, Naturalised Reptiles and Amphibians of the World. Essentially, it appears that they came over in his written record as eggs to a garden nursery in Bedford, at about the turn of the century. There is some debate over the date of that, but they were first heard calling in that area in about 1903. Unlikely that they came over as eggs, most likely as tadpoles, as has happened with other aquatic larvae of amphibians, of invertebrates and fish over the years. The reason being is that midwife toads are quite tricky to the eggs to raise away from the males. A lot of people have tried to do this in the past with some limited success. There's a whole host of controversy around it as well. Some of you will be familiar with the Austrian naturalist Paul Kammerer, who unfortunately ended up taking his own life in 1924 as a consequence of that whole debacle, but that's a conversation for another day. So it appears that the midwife toads came to Bedford accidentally as stowaways in a shipment of plants to a garden nursery. Wonderful, case solved. Particularly seeing, as we know, they probably came from France. So back in 2015, I went onto the MBN Atlas, the National Biodiversity Network. At the time, it was still called the Gateway. Now it's known as the Atlas. And you can see there are a number of blue splodges here where these records were over the course of 35 years. There's some there around Bedford, which is just east of Cambridge, basically ground zero. We've also got some on the North Norfolk coast, up towards Sheffield, in Suffolk, and then a load of splodges over in Wales as well. You know, lots of these are disconnected from each other. Just to highlight some of those, there's a big gap there from Cambridge up to the north there, or Bedford at least, also over to Oundle in Northamptonshire, over here in Howey and Newbridge-on-Wye. What's going on? We don't really know. We still don't know. We've got some potential answers. Down here in the south towards St Albans, High Wycombe, that area, what's going on? In West Runton, in Sheringham, in those areas in Norfolk, who knows? And then most importantly, Cambridge, which is where I was living at the time. I just finished my undergraduate degree at the ARU campus there. Now working for them. Funny how these things work. And we've been chasing midwife toads for a couple of years up until this point, until that summer in 2015, when we finally tracked down where they were. So we were using cool playbacks in order to detect the toads, and in order to find gardens where toads were, we printed off a whole host of flyers with my face on it and the midwife toad, and was like, hey guys, if you've got midwife toads in your garden, please email me or call me back and I'll come to your garden at night and look for these toads. Of 250 leaflets dropped off, only 3 people got back to us initially, which is still better than none, but it did mean that we were able to confirm that population was there. And the other important thing as well is that these residents were absolutely bloody amazing, because they kept me and my colleagues full of tea, coffee and cake until, you know, we were fit to burst, up until 2 or 3 in the morning sometimes, and they and their kids also got involved with the surveying and have done ever since, and it's been a wonderful community project as well that we've taken elsewhere outside of Cambridge. So we were using cool playbacks in order to try to locate the toads. Obviously that's biased towards the males because they're the ones that are doing the calling. If you've ever been looking for amphibians or any animal, you'll know if it's calling, you approach it, it goes stum very quickly. So we were trying to imitate arrival, get the males calling again, and hopefully get them to reveal where they were so we could then capture them, place them into individual Ziploc bags, similar to what Andrea showed us earlier with the Darwin's frogs, weigh them, measure them, and we also took buccal swabs from them, so just stick a swab in their mouth. Prior to COVID, kind of describe that as, CSI midwife toads. Now, everyone's been there and done that, so you're all familiar with the process. And we're also only targeting 5 to 10 individuals per population. We took dorsal photographs of each individual before release. We can try to track them through space and time, work on population estimates, and then we also sequenced the DNA of those individuals that we caught in order to then compare them to an online database on GenBank in order to determine whether or not they did indeed come from France. So with the Cambridge toads that we did this first in 2017, they all matched perfectly to samples of other midwife toads in Asturias in northern Spain, which is obviously different to what the literature told us about midwife toads came from. So this, kind of got us thinking, what the hell was going on with this population, and were the other populations the same, or could there have been another introduction pathway? So back in July 2019, we launched a kick, a crowdfunder project, We didn't have access to Grey Sports Almanac, unfortunately. That isn't the only Back to the Future reference in this talk, don't worry. So this is a quick map indicating where each of those populations were in the summer of 2019. The red ones were ones we knew about, hadn't sampled yet. Green ones are the ones we had sampled. And then the yellow triangles are ones that unfortunately were confirmed to be extinct. You guys can probably see where this is going. The next year, COVID hit, and we discovered a ton more populations around the country because everybody was restricted to their gardens, and they heard this unusual beeping sound coming from the bushes or near their ponds, and that's how midwife toads let themselves be known. These people would worry what it is, search online, see my details, get in touch, and so we were able to then confirm other populations, but also, most importantly, confirm that the Wandsworth population here in London isn't actually extinct, and we have since sampled that. Things have only got insane, even more insane since. I've put here over 20 populations have been buccal swabbed. The reason being is it's closer to 40, but I haven't actually told you how many we've been able to do that for yet. Unfortunately, because of time constraints, we haven't been able to use all of these samples in our analyses, but we do have some interesting results from the DNA from those different populations. So this is a haplotype network. Some of you are probably thinking, what the hell is this spider web of colours in front of me? We're using a similar colour scheme to that original diagram I showed you earlier, and all of those green blobs are those Spanish sequences that were closely related to the Cambridge population. So you can see the Cambridge one at the bottom there. All of the other populations in Great Britain that we've sampled are identical to populations from France. So that indicates that, yes, the literature was correct about the origins of these toads, but Cambridge is a little bit different. There's been a different introduction pathway there, and that isn't surprising given the fact that midwife toads were a popular pet at one point, but also a laboratory animal, and, you know, if they stowawayed once, why can't they do it again? You know, there could be a different thing going on there. So, as a summary there, all of our samples, except from one, matched populations, and there's some place names there of the locations in France. There's different sequences of DNA. I'm not going to try to butcher them because my French isn't the best, despite having a French surname. I know it's a travesty. The outlier was Cambridge, which we sampled first, which doesn't exactly match any single population as closely as the other ones do, but it has a close affinity to populations in the Pyrenees. This is an area of high genetic diversity in amphibians in Europe, mainly because during the last glacial maximum, it was a stable environment, and so there's more research that needed to be done to sample those populations to get a 100% match like we have with the other populations. So here's just a quick visualization of that. So this indicates the warmer colors. The red shows the match likelihood to those sequences of the different barcoding genes here. 16 there, so ND4 and CO1. You can see for most of those, there are large regions of France where there's a perfect match, but again, we're missing sequences here. There may be other diversity within there that we can then pinpoint to an exact location rather than an entire region, because as you are more than aware, hopefully, France is a pretty large place. So there's a large geographical area there that we haven't covered. Something also interesting is that we've been able to monitor these midwife toads over a number of years and see a whole host of other bits and pieces going on within them. So me and my colleagues, we described a number of limb malformations within midwife toads back in 2022. It's not surprising, given that these amphibians likely had very small founder populations. Their genetic diversity is probably very minimal. They're all very closely related to each other, probably, and this has seen some interesting malformations. The most interesting toad, please go and check this paper out because the pictures are amazing. There is a midwife toad somewhere with a fifth leg just sticking out of its chest like a chest burster from the Alien franchise. And hopping around Bedford to this day, which is marvellous. But also, as mentioned earlier by Andreas, thankfully, I think introduced this too much, we have the menace that is BD, the amphibian chytrid fungus. As they are an introduced species, we've been monitoring them for the chytrid fungi, and across 4 introduced populations we've found nothing, which is good. That's probably not surprising given they were introduced prior to the global spread of the amphibian chytrid fungus and those declines that were mentioned 50 years ago, but always good to keep an eye on that. midwife toads, particularly the common midwife toad, are one of Europe's most susceptible species to chytrid. We find them in large areas of amphibian abundance in people's gardens with 5 other native species, and so we're kind of using them as a barometer. If they're free of chytrid, then the other amphibians are likely as well, because as soon as they sniff the stuff, they kind of keel over. So we've been keeping that in mind as well. So just to summarise all of that, I know it's a lot to take in 10 minutes. God knows how your brains haven't exploded and squirted out your ears, but I'm glad they haven't. There are 2 distinct populations, or 2 different pathways, in Great Britain, and most of those populations are identical to those in Bedford and originated in southern France. We don't know where the Cambridge population yet has come from, but we are still working on this, and we have colleagues hopefully conducting some field work later this year to resolve those issues from the Pyrenees, and so far all of those British populations are free of BD. Now comes a little bit of a shameless plug for the British Herpetological Society that used to be based here at ZSL. I'm a trustee, and all of those notes I mentioned earlier were published in her bulletin. Rick Hodges, our editor, is pretty fed up of receiving notes from me on midwife toads, but it is what it is. Please go check those out, and if you like reptiles and amphibians, consider becoming a member. Also, I need to thank all these wonderful people, and Andy, our lab guru. I sent him some really poorly stored DNA samples. He managed to do all the lab work and save my bacon there, so cheers, Andy. But all of the other people there have provided us with the licenses, in Natural England's case, as midwife toads are non-native species. You can't just pick one up and release it again. You need appropriate licenses, otherwise you're breaking the law. But everyone else there, apart from my mandatory corporate logo there, has provided money to help facilitate all of this research. And we do have a paper in preparation at this very moment in time. So hopefully it will be submitted any day now. It's just finding the time to get that done. Unfortunately, I'm here today, so I would be doing it today if I could. But this just want to show you this nice picture of one of the Cambridge midwife toads. I didn't really share any previously, but they're a little bit larger sort of than the 50p piece. The males carry their eggs on their hind legs, and these are close to hatching. You can see the tadpoles are developing in there, a little bit of a yolk sac. And if you orientate properly, you can see the eyes of the tadpoles developing in the eggs as well. And my contact details are there if you have any questions. But thank you very much, and hopefully you found that enjoyable.
Excellent. Lovely. Okie dokie. Now we will hear from Lauren Maas, who will discuss the re-identification of Okapi in the wild. Lauren.
Thank you, everyone. It is a privilege to be here with you today talking about an extremely charismatic species. So this is a project that I have ongoing at the moment, and it's kind of in the preliminary stages, but I look forward to sharing with you where it is at. But first, some introduction about the Okapi. Many of you may already know this because we were lucky enough to have some Okapi here at the London Zoo, but they were not known by the Western world until 1901. They are the closest living relative to the giraffe, and they're currently listed as endangered with populations decreasing. There's an estimated 3,000 in the wild, and their home is the Democratic Republic of the Congo, which is seen here on the right-hand side of the slide. And numbers are reported to have fallen about 50% in the past 25 years. With that being said, kind of the motivation for this project was following a meeting that I had with the Okapi Conservation Project last autumn. They were telling me that it's really difficult in the Congo because it's really, it's densely forested where the Okapi live. And so they have trail cameras set up to monitor the Okapi, but it's really difficult to get that good estimation of the population. And they said, basically, they don't know if they're seeing the same Okapi 50 times or if it's 50 different Okapi. So we know that the striping pattern that they have on their rears is basically an individual fingerprint for them. However, for us, it's really difficult to discern the differences between them. So I had this idea, and I will state, fully disclose, I am not tech savvy, but I had this idea from other papers I had read. I thought, I wonder if we could train a computer to identify them better than the human eye could. So essentially what I did with these 4 beautiful creatures known to me at the Sacramento Zoo back home, I used them as a positive ID. So I took images of them, I have coined that the best day of my life, a great day in the field. So I spent a day with them just imaging them. I got about 3,500 images generated from video footage. These are some very beautiful photos of them, but I also got some that might kind of mimic what might be seen on a trail footage camera. And so from there, those images were fed into some models. And so the first model was very simple. It was just, can this computer ID whether there's an okapi in its field of view? Yes or no? So that was the first step. And then the second part of the model was sophisticating it a bit more to be able to tell if those okapi were different individuals. And so just some preliminary results with those 4 okapi. I'll play this little video if I click next. 3,500 images were sufficient to train the first model to ID okapi in varied circumstances. So whether they were out on exhibit, whether they were in clear view or obstructed, even with, like a viewing deck there behind, it was able to pick out, that's forest, pick out forest. And then the second model was successful at IDing specific individuals and telling them apart with 92% accuracy, which is fantastic. And the best part of this, potentially, is that it's something that's quite efficient and could be run on a cell phone out in the field. So future directions for this work, my hope is first and foremost to create a more robust data set. As many of you know, a sample size of 4 isn't that great. So I need some additional captive okapi images to feed into that. And hopefully the more images that we can give the model, the more sophisticated it will become. My hope is to be able to also make it more sophisticated and that potentially look into age and sex differentiation. We know that males have ossicones, as seen here, at a specific age. They develop those, and so that might be a means of telling difference between male and female. Gait analysis is something I'm also interested in. And of course, the overall hope for this is rooted in conservation. So hoping to be able to extrapolate this method for use in our Okapi there down in the DRC. So thank you. I know that was a whistle-stop tour. I had 5 minutes. I should have asked for more. But thank you, Dr. Zach. He helped with the machine learning because, as I said, that was not my space. The Okapi Conservation Project for the inspiration. Of course, Zoological Society of London for allowing me to be here with you today. And my friends and colleagues at the Sacramento Zoo, they were so tremendously supportive and enthusiastic with my ideas, both the humans and the Okapi. Thank you all.
Amazing. If you haven't been to see our Okapi here at the zoo, make sure you do. Oni and Mazimu, they're gorgeous. Anyway, thank you so much, Lauren. So within the fellowship, we are especially proud to have members of our Youth Advisory Board. It's a fantastic group of young people who play a vital role in advising ZSL and ensuring that youth perspectives are reflected across our work. So it was launched in 2023, and the board has really hit the ground running. And we're delighted to introduce members Grace Alagratam and Gaby Fadullon, who will be speaking with you today. Grace and Gaby.
Oh, and we've got Henry as well. Good afternoon, and thank you for allowing us to speak today. Hi, I'm Grace.
Hi, I'm Henry.
And I'm Gaby. And we're members of ZSL's Youth Advisory Board. The board consists of 12 passionate young people from various different backgrounds with the mission to unite and amplify diverse youth voices within ZSL and beyond, fostering an inclusive and equitable future for both people and wildlife. We are committed to representing and championing the voices of young people, not just throughout ZSL, but throughout the conservation sector as a whole. We're part of a generation that feels an immense pride, but also an immense pressure in being the planet's future custodians. We believe it's so important for young people's voices to be heard, as we know they can bring unique perspective, innovation, and passion if given a seat at the table. Being part of the board is an opportunity to take action, to change mindsets on environmental issues, to help get more young people into conservation, and to pave the way forward with hope instead of fear.
The board's been established since 2023, with the second cohort joining in 2025. Since July, we've been able to launch the ZSL Youth Conservationist of the Year Award, attend COP30, as well as spoken on numerous conferences. We've met so many young people who are just as passionate as we are. Did I mention we fed a few giraffes and 2 Okapia, so linking that back. It's been a whirlwind of a year for us. And as we've reflected on the past 200 years, we've also been imagining what's going to happen in the next 200 years. So we asked our fellow members, what do you want to see in conservation in the next 200 years? In the next 200 years, we hope to move towards something that's truly equitable, just and inclusive in terms of access for conservation. Conservation that engages directly with local communities and wildlife, both nationally and internationally. We would like to see an increase in pathways into conservation for all young people, and to see those in power act with the same agency that we know young people feel too.
We want to move towards a model of conservation that no longer focuses on crisis management, but stewardship, where care for the environment is no longer reactionary, but dependent on sustained systemic change in the way we grow food, use land, and harness energy. So looking forward to the next 200 years, the Youth Advisory Board hopes to open doors into conservation for young people, to bring youth voices into the heart of conservation policy, decision-making, and practice, voices that truly reflect the diversity of society. So if there are any time travelers from 2226 listening in, our message to you is this: the future we hope to build is the present we hope you are living in. We hope the seeds of youth action we've planted have continued to grow. Carry the legacy of all the amazing youth boards, activists, and allies that came before you, but the best is yet to come. If you want to find out more about any of the things we do, if you just want to have a chat, we have a stall outside, and we've also got a board game based on career pathways to conservation, so please do check that out. Thanks so much.
See you there.